So I'm Teresa Prztycka. I'm a senior investigator at National Center of Biotechnology Information, which is part of National Library of Medicine, which is part of NIH. And my group is working on a variety of computational problems in computational biology, and most recently, I think it's fair to say that we are focusing on system biology of cancer. And today what I'm going to talk about it is mostly a tutorial talk where I hope to introduce you to the new kind of growing area of interest that is of mutational signatures. So through this talk, I hope to explain what mutational signatures are, and specifically I also would like to tell you what are the computational problems that may be related to mutational signatures, and if time permit, we'll give some of the examples of algorithms and methods we've developed in my group. Okay, so as we all know, the DNA is the central molecule for life. It encodes information that is really necessary for cell to live, to reproduce, for organisms to survive, to provide through the evolution. So it's important that this molecule is being preserved, that it's not being mutated, that it's being stays intact. At the same time, it's really appreciated that DNA is really subject to constant attack from various DNA damaging factors. Some of it is, of course, environment, such as UV light yesterday, or various chemicals, pollution, drug, etc. On the other hand, also, even if we somehow miraculously have no external exposure, no exogenous exposure to exogenous processes, also regular processes that happen in the cells, cell duplication, gene expression, they all cause the vulnerability in DNA, and they all promote can cause DNA damage. It's been estimated that an average cell may have as many, may experience as many as 10 to the 5 lesions per cell per day. So that's an enormous amount of damage. And of course, we do not observe that damage because cells develop this very robust DNA repair mechanisms, which is very complex and also in many ways redundant. So most of those lesions are being very efficiently repaired, and we don't see them in the cell. Now, if we do see them, then most of them are, if they turn to mutations, most of them are those passenger mutations that are harmless, and only occasionally we may have a cancer driver mutation. However, another source of mutations appears when DNA repair mechanism is also faulty. This is also something that happens often in cancer, and then we have a combination of those environmental factors, the factors that are also endogenous factors, and they're all amplified by the fact that DNA repair is somehow compromised. Now, what's important to be observed, and this is not automutational signature as such as a relatively new field, but this observation is quite old, is that various specific mutational processes often lead to specific mutations. So, for example, UV light is known to cause a C2T mutation. But importantly, it's not that every C is equally likely to be mutated, but the context in the sequence matters. And if we consider the context, that's mostly this is the mutations from TC to TT. So the idea of with which people look at use the mutational signatures which are this pattern of mutation, is to, starting from those patterns, understand what are the processes that cause those patterns, what's their role in the cells, how cells react to them, or how also processes in the cells can lead to particular mutations. So informally, mutational signatures are just those patterns of mutations. In order to do anything with them, we have to define them formally. And I'm going to follow a very successful here definition of mutational signatures that's been mostly introduced by Alexander et al. in 2020, but similar ideas been around from other authors as well. So we start the definition of the mutational signature with something that we call mutational category. So we want to partition all the mutations that can happen in DNA into categories, and here the proposed categories is a triple. So a category is a triple of nucleotides together with the information that the central nucleotide is the one that is being mutated and what is mutated to. So this gives 96 different mutational categories, and I just wanted to mention that we really don't know. I say that C is mutated to T, but just looking at the DNA, we wouldn't know whether C is mutated to T or the complement of C, this is whether G is actually mutated to A. So we have 96 mutation categories, and then the mutational signature would be the distribution of the mutations across those categories and distribution of mutations that are actually attributed to a given mutagen. So for every mutagen, the mutational signature would be the distribution of the mutation across the mutation categories that are attributed to this process. Now, others, so I showed you UV light. There are just example of some other mutagenic processes. ApoBec is a really very famous enzyme that tends to that mutates DNA if it's in the single-stranded form. This has this characteristic signatures, and here we have mutations as the most frequent mutation will be mutation again from C to T, but in the different context. Smoking has yet another characteristic mutation, and many drugs, especially drugs that are being used in cancer therapy, have characteristic mutation patterns. Okay, so how we can discover those mutational patterns. Before I go, maybe I would like to maybe stop and motivate this a little bit to say why would we care? So of course, one important factor is that by studying this mutational signature, this will allow us to understand better the processes that in cancer, the processes that lead to cancer. It's also important for diagnostic prevention. So you would imagine that if characteristic mutational patterns are being associated with specific environmental exposures, you may want to uncover, by various statistical means, may want to be able to uncover those exposures that lead to the mutational signatures and then try to prevent it. And also in therapy, and this will be something I'll mention at the end. So I'm going to kind of use this figure from our review paper to discuss what are the challenges and what are the computational approaches and what are computational tasks around those mutational signatures. So the very first important task is, I just defined mutational signature as this probability distribution of the mutations that are attributed to a given mutagen. But really, we even don't know all the mutagens, and we don't know a priori those probability distributions. So it's possible, of course, that for some of those processes that we can uncover this patterns experimentally, like we could perhaps take a cell line, expose it to smoke, and see what pattern of mutation will emerge. But we can do it only for a very small number of processes. So the main idea now is to really use a computational method to come up with the dictionary of such patterns that are observed in human genome. So this is typically, although exact maybe model is maybe secondary, but usually we start with to uncover a mutational signature. We start with the large set of patients. So we have, say, N sample. Those are cancer samples. We have this 96 mutational categories. Perhaps you can also imagine some defined categories in a different way. And for every patient, we know the number of mutations that are in a given category. And now the goal is here to identify those signatures. Here are the matrix P, which are those probability distribution, and the matrix of exposures, so that the mutations in each individual patient can be explained as a linear combination of signatures. So the very first approach by Alexander for all was by using non-negative matrix factorization. You can imagine to achieve this task by other mathematical means. So what you came up with is, a set of the mutational signatures. The very first attempt produced first 5 mutational signatures, then 30. And this, as of June 2022, is almost 100 of mutational signatures. So here's a screenshot from the COSMIC database that contains currently known mutational signatures inferred through this non-negative matrix factorization. So, however, whenever people have a large consortia, when they have a new project, and when they sequence or analyze a new type of cancer, they will usually start also by identifying mutational signatures, see whether there are some new mutation signatures that should be added to this catalog. However, oftentimes you really, if you analyze a small number of genomes, then you are really not unlikely to uncover some new signature, but rather you would be interested that out of this catalog of known signatures, what are the signatures that were active in a given genome, and and what would be strength of exposure of the genome to each individual signature. So there's a number of so in this case, the problem is simpler. We have, again, this set of genomes and mutations in each category. And we assume now that all the mutational signatures are being known, and all our goal is now to decompose it to find the mutational signature out of those predefined set, which will explain the mutational profile of a given individual. So there's a number of methods that have been produced to do that. The very first one was the construct-seq, which was a very kind of simple one. Our contribution was also quadratic programming, where, in addition, we point that we really need to have some statistical significance of those mutational signatures, so that one. And then there's a number of new approaches, some of them using Bayesian approaches, etc. So this is, identifying mutational signatures that are present in a given genome. Okay, so once we identify those mutational signatures, perhaps another question that I would like to mention is what are the properties of those signatures? And this is a little bit, I think, important, so I am going to inject a little bit of biology here. But perhaps one of the important things that, as I describe you may think about it that whenever you see any triple of nucleotide any element of this from this mutational category, they're not actually equally likely to be mutated. So not only the immediate context matters, but also the larger context matters. And why it matters here? So for the processes, for the mutations that are being introduced because of biological processes in the cell, so the key biological processes that involve DNA molecule would be the replication and transcription. So in the replication, the DNA molecule is, two strands are being separated, and the copy, and from each individual strand, the double-stranded complementary strand is being constructed. And this process is asymmetric, so we have so-called leading strand, where this is relatively smooth, and so-called lagging strand, which the process is really going in a little intervals, leaving this strand that is the lagging strand in the replication open to mutations and more often. And also, another thing that is also important is related to 3D structures that have been mentioned by Christine, is that most of the genes residue in the regions that replicate relatively earlier. And therefore, cell recruits most of the repair machinery more efficiently at the beginning of the replication process than later. So there is much more errors in the later steps. They are repaired less efficiently than the beginning. Okay, now just to mention also that transcription is yet another important process where one strand is being transcribed, another is not. That that allows the strand that is not transcribed to be often more mutated. And finally, the general structure of the DNA molecule is, as you've seen in Christina talks, DNA is some of the areas, some of the fragments of DNA are really open, others are being, round about these nucleosomes. And then again, depending where this DNA is, it may be exposed more to the mutational processes. And also, depending on where it is, it may be easier or harder to repair. So that is just important to remember that there are this variability, and it's not all equally likely. So with that, for example, the question is like if you look at the mutation and assume that you even previously used one of these programs to uncover which mutational processes are active in a given cell, even what is the exposure of a given mutational processes, it may not be always very trivial to take a given mutation and being able to assign it to a particular process. So this is based on the hypothesis that neighboring mutations are likely to result from the same or related mutagenic process. A couple of years ago, we developed a hidden Markov model that will help to assign mutations to given mutational signature. This model assumes that the mutational signature are known, that we also know the exposure of each mutational signature from this, previous methods, and it's just our goal is to look at the mutations and try to assign them to one of those signatures. And the main idea is to divide the mutation into two groups, one which I call disperse, where two mutations are far away from each other, two consecutive mutations in the genome are far away from this other, and for this one, we simply model them as multinomial process, and the clustered mutation was that are nearby. This is the ones that we model with hidden Markov model. Now, I'm not going to go into more details here, but just to what I mentioned before, now we can look at here in blue are the mutations that are in this sparse, that are distributed across the genomes, that are not clustered. And what we see here is the number of mutations depending on the various signatures, depending whether this is early replication or late replication. And this is like emphasize what I just said, that for most of the signatures, you can see that you have more mutation in the late replication regions versus the early replication regions, except in red are those mutational signatures that are restricted to those clustered regions, and those behave sometimes differently. Okay. So this is about the processes. Now, perhaps computationally, most interesting question, at least for us, was that, okay, we have those mutational patterns, and really for most of them, we know very little what are the causes. I mean, I should say that it's always, if you depend whom you ask, some say we know a lot. Well, glass is half empty or half full. So how do we know if since those mutational patterns are being uncovered in a computational way, how do we find the association between those mutational signatures and specific mutagenic processes? The whole idea of coming with this mutational signature was the appreciation that there is this that was initially being hoped to be a one-to-one relation between mutagenic process and this mutational pattern, the signatures. Well, first of all, as I say, in some examples, we can have experimental study that can, when we can directly measure the response of the cells to a particular mutagen. Most of the results are based on the associations. Namely, we observe that signature 4 is associated with smoking, and we call it smoking signatures. It's not only associated with smoking, but it's correlated with the amount of packs. That, for example, to signature 1 and 5, that been observed to be related to age, so we call them clock signature. And there's a number of others that very, by this type of association-based approaches, have proposed a specific mutagenic process that's related to the signature. However, as another problem is that even if we associate a mutational signature with a process like age, this doesn't explain the mechanism. And also, many signatures have unknown etiology, even any sort of etiology based on the association. So this kind of leads us to the question how we can link the mutational signature to the causes. And I'm going to maybe mention two approaches. I'm just briefly maybe skip about the third. So, well, we have to use the additional data to help us to suggest what mutational processes may be associated with given signature, what biological processes may be associated with a given signature. And what would be the additional data in cancer that you may think about it? Well, here we just consider that, that we know the gene expression, and we'll try to understand how gene expression relates to mutational processes, to mutational signature, and maybe that relation will be informative. Another question, what we haven't asked yet, is about methylation, and probably we should have. But yet another question is, most of this analysis is done in cancer, so how cancer driving mutations are related to those mutational signatures. So the very first one is, of course, the expression. So for doing that, we usually focus on a specific cancer individually, and assuming that we have for the same set of patients, we know mutagenic processes that were acting on a given individual, and what's exposure or in other words, strength of those mutational processes, so that we have one matrix patient per signatures. And at the same time, what many of you probably used in this, people who work on cancer used in their study, is the expression data, where we have another matrix that for every gene, we have gene expression across patients. So now we can compute that for every pair, gene and signature, we can ask if there is a correlation between gene expression and strength of the signature across the patients. Keeping up only the genes, so that will give us this matrix, genes per signatures, where color-coded here is association, positive or negative. We can filter out some of the genes that are not significantly associated with any of the signatures. Then we cluster the pattern of signatures, the genes that have the same relationship with the signature together, and now once the thing is clustered, we can use co-enrichment or other type of analysis that will help us to understand what those clusters represent. So when we did it for breast cancer, and kind of both we can both appreciate that's very useful, but also has some limitation. What's very useful, so here we have again the signature. Here we have clusters. Okay, sorry, I'm losing this. Here we have clusters, and here are co-enrichments of those clusters. So obviously, from this one, we have a bunch of signatures that are very correlated with each other and are correlated with the same expression of the same activity of the same pathways. And we wouldn't be able to kind of say anything more detailed. However, the two clock signatures are really showing different patterns. And one signature 1 is associated with cell cycle, and that's consistent with what we know about the signature. This is corresponding to mutations that are being collected at each replication. And this is kind of interesting for the people who are interested in timing the evolutionary trees and other processes, namely this early signature, the strength of the signatures counts the number of cell cycles. So that's really useful. And the other signatures that is also related with age, related with time, doesn't do that. Instead, it seems to be associated with specific DNA repair processes, which are really very main repair processes that repair lots of environmental exposure. So we assume that the second signature is simply age-related signature that relates to all the exposures that we collect through the years, and yeah. So this is just the summary, that we have those two clock signatures that seems to be clicking to different clocks. One is the cell cycle clock, another is kind of, I'm thinking about it, walled clock, which is potentially related to reactive oxygen species. So as some example of applications, we can recently kind of those in the context of COVID, there was lots of interest in understanding how smoking can affect, severity of the COVID diseases. So we've been motivated by this question, but this is and ask a different one. Say, okay, can we use mutational signatures as the sensor of environmental exposures, and use the gene expression data to see how smoking influence say cell type composition in lung, and we could uncover, and this just briefly summary, that we could see that smoking signature 4 is related to increases of certain cell types in the lung, while decreases some other cell types. And without going into the details, it gives us some insight how smoking can lead to lung damage. And the second here application, this is I wanted to mention, is this opportunity to use signature 1 to model time. So here this is from paper of Genstruck et al., where they try to, using the exposure to signature 2, are trying to estimate how far before the patient came to the clinic with cancer when the tumor started, how many years before the tumor started. And the idea here is that basically you have a tumor being growing with that you have cell division consistent with a given tissue, and then when tumors start to develop, you have much acceleration in the cell division. So this is how the mutational signature 2 is being acquired. Okay, so as I said, we're going back to what we've been able to achieve by the clustering, and this glass is half empty, half full. We could learn something about those two clock signatures, how they are different, but there is this blob of signature 13, 3, and 8 that we cannot dissect. What are the molecular pathway each of them is associated with, because they kind of are all associated with each other. So for that one, we have to forget the clustering and try to have a more refined process. And here, a postdoc in my group, Bayabatar, came up with the idea to use what perhaps many of you have been exposed to, is to use a process similar to that we find a regulatory network, but to understand what is the relationship between signature in individual genes. So here we assume that as we know, gene expression we can consider so for every patient we can consider we can measure the activity of a given gene. Here, similarly, we can do similar things for signatures, because we know the exposure of a signature. So for every patient, we can measure also the activity of the signatures. So we have two different types of nodes, but after normalization, we can think of them that we have genes and we have the signature, and for every patient, we have activity of either of them. And then we can use the network inference method to try to capture the relation between nodes and signatures. And here, we construct a graph. You can see a directed graph. You don't see any more node of signature because computation doesn't matter. But we start with the gene expression for the gene, signature exposure for the signatures, and then the idea is to use sparse partial correlation techniques to explain the expression of each gene as a linear combinations of gene or signature, as a linear combination of other nodes, which could be also gene and signatures. So maybe let me skip. Oh, okay. So in addition, okay. Let me skip this one. So this is additional steps that perhaps is not so important here, but just I wanted to. From the statistical perspective, we also look at second method kind of techniques to allow to identify, if possible, whether the information flow is to disambiguate the direction of the information flow, which sometimes we could do, sometimes not. Okay, so that's. So this was just to say that this additional step improve over other methods that were trying to infer the relationship between activity of nodes in the network. So okay, so this one now give us a network that for every signature, so we can see what are the genes that influence those signatures, or at least up to the. And which genes are being influenced by the signatures. We can now, this is again the same breast cancer data. Now we can appreciate that between two signatures, the information flows from the signature for the homologous recombination signature to apobex signature. So we kind of untangled this chicken and egg problem in the previous one. We can also see that for one signature 8, for which we couldn't say anything, now we can say that we know it's associated with nucleotide repair process, which has been also shown later on experimentally. And we kind of get a lot of information here. From the perspective of a computer scientist that analyze this data, this makes it a little bit more difficult because it's by this specification we have some kind of limits our ability to use like Go enrichment and go after typical gene enrichment analysis. Nevertheless, it was still very informative. Okay. So the other things, so one thing is to look and see how those mutational signature may be related to gene expression, and I didn't dwell on specific biological processes, but you could appreciate from what I say that for some signature you will be able to pull up the pathways that are associated with a given signature, and at least make hypotheses what are the causes, or at least associated processes with a given signature. Another thing would be, of course, this is a cancer. So, the focus, that's important for each cancer is to appreciate that there are those cancer-driving mutation, cancer driver, and it's reasonable to assume that those cancer drivers are not neutral when it comes to mutational signatures. And they are not neutral in two different ways. So the first question is, are mutational processes lead to specific cancer driver mutation? And the second direction is whether, sorry, that's the same direction. That was not meant to be. But the second direction, whether cancer-driving mutation, specific cancer driver mutation, may be associated with particular signature. So both of those questions have been asked, and as the data will continue to accumulate, we may be able to be more empowered to ask this question. But there are examples that some known mutational hotspots have been associated to specific mutation signatures, and that helps to explain occurrences of some cancer drivers. And also in the second direction, this is something that a method that we developed. Now, what in this case, when you ask the question whether any cancer-driving mutations influence may contribute to emergence of particular signatures. So this is a similar problem that what I've been seeing in a population study. Namely, we think of the signatures of the mutational process as a phenotype, and we ask whether there is an association between this genotype and phenotype. So the formulation is similar, although many approaches that are developed to cancer are not necessarily cannot necessarily be used in the population studies, mostly because cancer is really very aggressive diseases, and these phenotypes are kind of more pronounced. So this is to say that often this is easier. In this case, we develop a method that for every patient we have this continuous phenotype that is a strand of the signatures. I just color it here that strand of the signatures sorted the genomes by the strand of the signatures. We also have for every individuals we have the gene alteration. Here we do not take all the passenger, but we take only the genes that are frequently mutated, so potentially drivers. And in addition, we take information about the relationship between genes, that are known interaction between the genes. And this is being assumed, this is, starting with the assumption that if you take two genes that are interacting with each other, a mutation in any of those genes may have similar effect on the cell. So then we formulated this one. This is not machine learning. This is an optimization problem, an integer linear programming problem. So we formulated this as an optimization problem when we would like to find optimal module so that we can, the mutation in the genes in this model can explain the enrichment of the signatures in the patients, in the stop patients. So as linear model, this is maybe the formulation. I am not going to go into details, but just for people who may have not seen the integer linear programming, this is really a very convenient formalism to express many biological problems where you express the problem as an optimization problem. Usually you would like to find, say in this case, you would like to find a set of genes which will optimize the sum of weights, so that meaning that they will be the weights are related to how strong this phenotype is. And then what it makes it, eventually we say that we want this to be a network. So this is being formalized by the number of additional constraints, the linear constraint. And if you are able to express your problem in this way, then if the size of the problem is not really very big, you may be able to solve it optimally using existing integer linear programming solvers. And in this case, we have been able to come to this optimal solution. And then for many of the signatures, we could identify these modules or the subnetworks that are associated with that signatures. Okay, next thing's therapy. So that's something that is still, I would say, a little bit underdeveloped. But the very famous example is that we know that breast cancer patients benefit from a particular drug, PRP inhibitor. And that being then, I associated that, really the patients that have specific signature 3 are really benefit with this from drugs from PRP inhibitors. And then from that observation, we can go further on and say, okay, what other cancers have this signature? Ovarian has, and it was see that, okay, ovarian cancer, if patient has the signature, will also benefit from this therapy. There are some other associations. This is all by association here, so mechanism, there's a little bit understanding of this mechanism here. But the next two are mostly by association, is that patients that have the ApoBec signature 2 and 13 benefit with immunotherapy and is being even approved in some cancer types. And also signature 4, this is the smoking signature, so maybe a little bit contradictory in the sense that smokers that have a lot of the signatures will also benefit from immunotherapy. So that's one way of thinking about it. Last thing that I would like to go is to re-examine the models that we started with. So we started with the assumption that all these mutagenic processes are additive, right? Because we started with this matrix of the mutations patients by mutational count, and say we would like to explain the mutation in each patient as the linear combination of the signatures. Well, that's very nice and very simple and elegant way of doing that, but it's not fully correct. So as I mentioned, it really takes two to tango here, and the process is a biological process in which, as a result of which we have this mutational signature, has two steps. One is that we have DNA damage, and the second is DNA repair. If this DNA repair process were always, perfect, that's fine. But in cancer, one of the problems is that this is this DNA repair process that is also being broken, and then we have some signatures that are associated with DNA repair, but that here we start to have a problem that we have this explosion of signatures. In a way, you can think about it that we should think about it that these two steps that we have. First step would be DNA damage. The second step here is DNA repair, and what we observe are those signatures on the outer cycle. So, there's so many ways, different ways things can go wrong. There's so many interactions that between DNA damage and repair. So no wonder we have more and more signatures. And that's getting to be a problem because we have lots of signatures, it's harder to decompose them. So maybe we should go back and revisit this issue and say, should we have really additive model or, consider some other ways of modeling of the mutational signatures. So in Recomp a couple of years ago, we proposed a models that is, rather than being linear, it relies on the composition of two types of mutational signature. We divided mutational signature as primary, that we think about them as those environmental signatures, those are that causing damage, and secondary as typically related to DNA repair. And then, for the primary signatures, we use additive models like before, but we just assume that to model the secondary signatures, we assume that the secondary signatures, the DNA repair signatures, act on the results of the primary processes. So we, what? Okay, yeah. So I have 45. Okay. So this is we call repair 6. Again, we could identify the model those separately, these primary and secondary signatures, and the question is, did it help something? So in the breast cancer, in the breast cancer, which was actually our motivation to look at it, there were 3 signatures that are associated with mismatch repair. So we hypothesized that these 3 signatures are really occur because we have 3 different, maybe I should go back to this one, because we have 3 different signatures that are being modified by mismatch repair signatures. And indeed, when we apply our models, we could replace all the 3 signatures by 1. So you could say that indeed, the composition helped. Now let me skip this one. So this is just simple checking that this smaller number signatures are better and etc. And in fact, also we have a validation that the mismatch repair signatures that we identify as the only one is consistent with the, so this is the mismatch repair signatures that we identify into different versions of the approach, and is consistent with the experimental signatures. Okay, so I guess that's I will conclude with this one. So this kind of summary slide that we started with the introducing to the mutational processes as being kind of a composition of both environmental processes, the exogenous processes that are related to biological processes that happen in the cells, and on top of that we can have processes that are related to dysfunctional DNA damage. And as comes to the computational problems, the first one is actually identify those signatures, and here the example was using non-negative matrix factorizations. Once we have the signatures, it's interesting to go back and to understand, to link them back to biological processes in many different ways, seeing what are the properties in terms of, are they uniformly distributed over the genome, or they are maybe a kind of focus on particular genomic regions, and then, looking at using additional data such as mutational data or gene expression data, trying to understand what's the relationship between those mutational signature and biological processes, and that helps us to link some of the processes, some of the signatures of unknown etiology, at least to hypothesize the processes behind it. And finally, I just pointed to a link to therapy, and this is still mostly by associations, but this is, I should say, a little bit another line, possible line of studies. And as for machine learning, I think there's still lots of opportunity to approach this, adapt some of this machine learning technique to the questions related to mutational signatures. So what are the open questions? So I kind of at the end just briefly mentioned that the assumption of the additivity of signatures is oversimplification. I suggested one of the composite model. However, this is just first model along these lines are trying to be nonlinear model, and there's much to be done to consider other type of other ways of modeling mutational signature that will be nonlinear. There is the question now when we have so many of those signatures, there's 100, and more are coming, is that what we are going to do with them, how we can classify them, or maybe can we at least even understand that whether a given group of, say, signatures is associated with related processes, which are related, which are not. So this is not much being done. There is also more methods to relate this signature to biological processes. We tried two, but more things, more methods would be interesting. And then, of course, we as the data increase and we have more and more data, we know that may enable this question, also maybe introduction of more, say, machine learning type of approaches. Okay, and I would like to acknowledge my group, current and former group members, funding from the Intramural Research Program, and some of the collaboration of our mutational signature studies. Thank you.